As we learned in the previous module, neurons are electrically excitable, which means they communicate with one another using an electrical signal. The generation and propagation of these electrical signals is possible due to the distribution and movement of charged ions within the cytosol of and the extracellular fluid surrounding a neuron. So an ion is an atom that has a net positive or net negative charge. For example, the ions calcium, sodium, and potassium all have positive charges, whereas phosphate and chloride ions are both negatively charged. Within the human body, ions are located in both the fluid within and outside of cells. If we take this image here, this blue is the extracellular fluid on the outside of the cell. This here is our plasma membrane, and this yellow is the cytosol or the fluid within the cell. Floating around in both the cytosol and the extracellular fluid are ions. You can see those represented by all these different shapes. The negative or the positive sign within those shapes is representing whether these ions have a negative or a positive charge. So the generation of a nerve impulse, which is the electrical signal that a neuron uses to communicate, is possible because of two important features of the plasma membrane: the existence of a resting membrane potential and the presence of specific types of ion channels within that membrane. So resting membrane potential is the difference in an electrical charge across a membrane. The presence of specific ion channels within the membrane means that the ions can move either into or out of the cell, down their concentration or electrical gradient, which, remember, is that difference in the concentration or charge of one region to another. When ions move across the membrane, either into or out of the cell, they create a small electrical current. So considering this information, now looking at this image, and the resting membrane potential is established due to the difference in the net electric charge of the ions on the inside of the cell compared to outside of the cell. So for example, if we added up the net electric charge of all of these ions in the cytosol, it would be a net negative charge, and you can see that represented by these minus signs here. If we added up all of the charges of the ions on the outside of the cell, there would be a net positive charge, and again you can see that represented by these positive signs here. So this is the resting membrane potential, or the difference in the net electric charge from the inside to the outside of the cell. We measure this difference as voltage using the units millivolts. Now, because of this resting membrane potential, or the difference in the electrical charge from the inside to the outside of the cell, when these ion channels open in the plasma membrane, the ions are going to want to move down their concentration or electrical gradient, either into or out of the cell. This movement of ions through those channels is what creates the electrical current. Now, the specific ion channels found along the membrane of a neuron come in a few different types. First, we have leak channels, and these channels randomly alternate between open and closed positions, letting the ions specific to that channel slowly leak into or out of the cell. Typically, in a neuron, there are a high number of potassium leak channels and a small number of sodium leak channels. Next, we have ligand-gated ion channels, which open and close in response to the binding of a ligand or a chemical. A wide variety of ligands, including neurotransmitters, hormones, and particular ions, can be the stimulus for the opening or closing of these ligand-gated channels. The neurotransmitter acetylcholine, for example, binds to the sodium channels on a muscle to allow for the influx of sodium, which then in turn causes a muscle to contract. Ligand-gated channels are found in the dendrites of most of our sensory neurons. Receptors like nociceptors or pain receptors, as well as interneurons and motor neurons. Ligand-gated ion channels also make up part of a chemical synapse, which we will speak more about shortly. A mechanically gated ion channel opens and closes in response to mechanical stimulation. So this could be in the form of vibration, such as a sound wave. It could be touch or pressure on the skin, or stretching of a tissue. The mechanical force distorts the channel from its resting position and opening the gate of the channel. Examples of mechanically gated ion channels are those that are found in the auditory receptors in the ear, the receptors that monitor the stretching of internal organs, such as the organs of the gut, and in touch and pressure receptors in the skin. Lastly, we have voltage-gated ion channels, which I'm sure you could guess, open in response to a change in voltage or a change in that membrane potential. A voltage-gated ion channel will open when the membrane potential hits a particular change or voltage that we call threshold. Voltage-gated ion channels are particularly important for the generation and propagation of action potentials in the axons of all of our neurons. And we have specific sodium, potassium, and calcium voltage-gated ion channels in these regions. Speaking of voltage-gated sodium channels, and these channels work slightly differently to other voltage-gated ion channels, which simply open and close. Our voltage-gated sodium channels have 3 different states that they can be in. They can be closed, like any other ion channel. So during this time, no sodium ions can move through the channel. In this state, the ion channel is closed, but it is capable of opening. Why that is important, you will understand in the next learning objective. We then have open, which again is like any other ion channel, where when these channels are open, the ions will move through the channel down their concentration or electrical gradient. And then we have inactivated, which is a state the voltage-gated sodium channels move into after being open. And in this state, the gate on the ion channel is closed, so ions can't move through the channel. But from this inactivated state, the ion channel cannot be opened. Now, we will come back to this again shortly, but what I want you to remember is that our voltage-gated sodium channels start closed. With a change in voltage or membrane potential, they open. They then become inactivated before they again close. Now, one more specific channel or mode of transport that I want to speak about before we move on to the next learning objective, and that is our sodium-potassium pump, which we did mention briefly when we spoke about the transport of substances into and out of the cell in the cells module. And relating to the nervous system, sodium-potassium pumps are important for the restoration and the maintenance of the resting membrane potential. Remember that if we don't have a membrane potential, in that if we don't have a difference in the electrical charge between the inside and the outside of a neuron, then the ions won't move across the membrane when the ion channel's open, and an electrical current won't be generated. So how the sodium-potassium pumps work, they first open up into the cytoplasm, so in the inside of the cell, and 3 sodium ions will bind to the pump. The binding of those sodium ions causes the breakdown of ATP, or energy. The release of this energy causes the pump to change shape. Now the pump opens up to the outside of the cell. It releases its 3 sodium ions into that extracellular fluid. Once the sodium ions have been released, 2 potassium ions can now bind to the pump. This causes the release of the phosphate ion, which was left over from the breakdown of ATP. The release of that phosphate ion then causes the pump to change back to its original position, so opening up into the inside of the cell. The potassium is now released into the cytosol, and sodium can then bind again. As I mentioned, sodium-potassium pumps are essential for restoring resting membrane potential following an action potential.